Hello everyone and welcome to this presentation where I'll be talking about my latest paper on generative AI for drug discovery. I am Omar, a technical lead in AI engineering at IBM Morocco. I'm also a researcher in computational drug discovery. So, um, drug discovery is a very important part of the whole process to deliver a drug into market. Drug discovery alone could take up to 10 years sometimes, and delivering a drug to market could cost roughly $1 billion with all the clinical trials, clinical studies, and preclinical studies and drug discovery. So we wanted to introduce AI into this part, the drug discovery part, in order to accelerate it and find more optimal ways to achieve it. So what came into mind is the study of protein-ligand docking. So, given a protein structure, a 3D structure of a protein, and a ligand could be any small molecule. Um, the goal is to predict the binding site and predict which ligand has the minimum binding energy. So, why are we looking for minimum energy? So, think of it this way: if you have a macromolecule target and a ligand, you could bind that ligand to that um, to that target given an amount of energy. So if you give X amount of energy to the ligand, it could bind to the target. But if you have a negative energy, meaning below zero, the ligand binds automatically and it diffuses that amount of energy. And if you have, for example, -6, it's better than -5. And we're trying to find the ligands with the, the most minimum energy possible. To calculate this binding energy between the macromolecule and the ligand, we used this software which is called PyRX. It is very handy to calculate for estimating and calculating the binding energy of a ligand with a macromolecule. And we wanted a use case where we could leverage this power of Gen AI in oncology or cancer therapy. So one of the most critical targets in oncology today is the epidermal growth factor receptor, which is a transmembrane tyrosine kinase receptor, and it plays a great role for cell proliferation and survival and cell growth. Sometimes these genes in some form of cancers like non-small cell lung cancer and colorectal cancer, glioblastoma, which is a brain cancer, and some other cancers like breast cancer, this gene is overexpressed or mutated or dysregulated. So if we inhibit this its protein, we could theoretically stop the advances of those said cancers and give better chances with chemotherapy and or radiotherapy. So we went for our ligands. We used a data set available online called the Symbiotic data set or KMBL data set um, in a smiles representation. So smiles is a type to encode molecules using only text. So we went for, as you can see in the image here, there's a lot of other representation techniques for molecules, but we went for smiles or canonical smiles because one, they're a sequence and they're unique and invertible. So meaning if you have a molecule A, it will have a representation in canonical smiles A, and so there is only one representation, and each representation can only represent um, one molecule. So the data set has nearly 500,000 small molecules that are already used for therapeutic use cases. So those are 500,000 pharmaceutical compounds in a SMICE format. So for the generative AI models, we went with a GPT-2 based model, and we did a study. So we tried to use a GPT-2 model along with an LSTM model, and we compared the results to see which model is gonna give better results. Yeah, we trained the model on the smile sequences, those 500,000 smiles. We fine-tuned the model for validity. So when we're talking about drug discovery, there's there is some other metrics that are relevant, uh, not only the classical metrics like accuracy or loss, but there's some other metrics like validity. So we could have a model that generates molecules, but there could be some invalid molecules. Like we know that, for example, a carbon atom has 4 bonds, but for some reason we could generate a molecule where a carbon atom has 5 bonds, which is invalid chemically and doesn't obey to the laws of chemistry and physics. So that molecule is gonna be void. um, this is why we are introducing these new metrics to better evaluate these generations and these models. So, of course, we want a model with a high validity rate. And there's also another metric, which is uniqueness. We don't want the model to generate the same molecule twice. So we are looking for duplicity and unique unique molecules. The goal here is to train a model first that could generate smiles. Um, Once we achieve that, we generate a number of presets of smiles, like 1,000 smile or 2,000 smiles. After that, we process those smiles. We remove any duplicates, we remove non-valid molecules, and we filter the molecules. I'll explain further what are those filtering criteria. We pass afterwards those molecules into the tool that we talked about, sorry, PyRX, which will give us the the binding energy of each SMILES with the macromolecule that we used for EGFR. We then we will choose the top 100 SMILES, for example, that achieved a low binding affinity score, and we're gonna use them to fine-tune the model so it can produce SMILES this time that are similar to the ones that had scored best. So talking about the filtering and evaluation of those molecules. So there's a rule in pharmacology. Um, this rule said that for a drug to be a good pharmaceutical drug, it has to obey to the Lipinski rule of five. So the Lipinski rule of five states that a good molecule is a molecule that has a molecular weight less than 500, a lipophilicity or log P lower than 5, an H bond donor lower than 5, and an H bond acceptor lower than 10. There's also another filtering metric that we used, that's synthetic accessibility score. Since we're dealing here with novel molecules, so molecules that aren't manufactured yet, so we didn't want to introduce molecules that could be hard to manufacture. So this synthetic accessibility score will give us a score between 1 and 10, 1 being the easiest to manufacture and 10 being the hardest. And of course, we want some molecules that are between 1 and 3, so it can be easier afterward to manufacture. These are the results from these are the results and the analysis between the GPT-2 model and the LSTM. As we can see here, the LSTM performed very, very well against the GPT-2 model. So we went with the LSTM to produce our results. As you can see here, the LSTM had a validity rate with 98% against GPT-2 model based model 53%, which is very low. So if we, for example, if we generate 100 molecule, the LSTM model will produce 98% or 98 molecule valid, whereas the GPT-2 model will only produce 53 molecules that are valid. So these are some of the existing EGFR inhibitors, like erlotinib or afatinib. So erlotinib, for example, has a binding energy of minus 7.5, and the lowest available on market today has a binding energy of minus 8.7. These are our results along with the pharmacokinetic properties of these molecules that we generated. And as we can see here, the results vary from minus 9.4 to minus 10.4, which surpasses the existing EGFR inhibitors by an order of magnitude higher or lower. So yeah, that was the whole core of the paper. But I invite you to read the paper. I could leave the link in the description if you want to read the paper furthermore for more details. Yeah, thank you.